Okay, welcome. Thank you for everyone for coming. Great to see so many faces in the Douglas Hall. Welcome also to those joining by Zoom and also those joining from Zoom from Munich. So welcome. This is the special seminar series presented by the McGill Douglas Max Planck Institute of Psychiatry International Collaborative Initiative in Adversity and Mental Health. So my name is Jermaine Lowe, and I'm the AMH coordinator. So we call this actually the AMH initiative, sorry, because it's so long. So as the name suggests, it's a partnership between the McGill Douglas and the Max Planck Institute of Psychiatry in Munich. And what it aims to is establish a world-renowned, world-leading research and training program that studies studying how early life adversity is biologically embedded and how this increases the risk for psychiatric disorders. So if you want to learn more about it, please visit our website. It's actually adversitymentalhealth.com, or you can email me at coordinator@adversitymentalhealth.com. So I'd like to also mention that this session is being recorded, and the recording will be available to view at a later date on the Douglas Research Center YouTube channel. So, and also, please, if you have any questions, keep it to the very end of the talk. We have a designated time for question periods, and also the people on the Zoom to ask questions. So without further ado, I will pass the floor to Dominique Walker.
Thank you, Germaine. It's a real pleasure and honor to introduce Dr. Tali Baram, Distinguished Professor of Pediatrics, Anatomy, Neurobiology, and Neurology at the University of California, Irvine. Dr. Baram is an MD PhD, and since 1995, she holds the Danette Shepherd Endowed Chair in Neurological Sciences at UCI. She is the current director of a large Conti center at UCI that is focusing on early development trajectories, and in particular, with respect to fragmented early life environment and its consequences for adult psychopathologies. Dr. Baram has established one of the most currently used animal model of early adversity worldwide in the form of limited bedding and nesting material paradigm. And I know that some of us are using it also here. She has greatly contributed to our broad understanding of some of the critical mechanisms leading from early adversity to adult psychopathologies. She has demonstrated, for instance, that the unpredictability of maternal care or parental care can influence synaptic connectivity in key brain nodes associated with stress responses and emotional circuits. And in fact, if you haven't seen, she has an editorial, very beautiful editorial piece in Science in the last June 3rd, I believe, right? So I would recommend you go and check it out. So it disrupts, early adversity disrupts hormonal and behavioral responses to stress and might lead to anhedonia and altered emotional processing of threats. In addition to her internationally recognized work on early adversity, and she's equally well recognized for her work on epilepsy. Founder and director of the UCI Epilepsy Research Center in 2002, she recently demonstrated that prolonged seizure-like activity caused an overall drop in specific microRNAs levels in the hippocampus, and that is caused by a large release of this microRNA from neurons via extracellular vesicles. So a topic that is very hot right now. She has also revealed that in a model of temporal lobe epilepsy, suppression of glial neuronal dysfunction might hold promise for mitigating seizure-induced hyperexcitability and epileptogenesis. For both fields of research, Dr. Baram received numerous awards and lectureship nominations, among these the prestigious George Coetzee Lectureship from the American Academy of Neurology, which is the highest research award from this society, the Bernard Sachs Distinguished Award for Research from the Child Neurology Society, and the Javits Neuroscience Merit Award from the NIH. She is also an elected fellow of the American College of Neuropsychopharmacology. Throughout her career, Dr. Baram has assumed many leadership roles as a scientist, chair of executive boards, and as a mentor. This has never been to the detriment of her outstanding productivity, with publications appearing regularly in the top journals in the field. So I don't want to take too much time, but I also would like to mention that in addition to being an outstanding scientist, Tali has been a wonderful friend and mentor throughout the last 30 years, I believe, of our respective careers. Her excellence in research has sacrificed nothing to her extremely generous human qualities. So please join me in welcoming her to the podium.
Sometimes I wonder if who is the person she's talking about here. I have a very soft voice, so I hope that both the folks in Zoom and the folks in the room can hear me okay. Well, it is really a very, very special pleasure to be here. First of all, I find the remarkable international collaboration between two of the world's best places to study the origins of mental disease quite remarkable. And I'm really honored and thrilled to be here to tell you a little bit about how early life adversities shape adult behavior, including psychopathology. This is particularly exciting because together the McGill-Douglas Center and the Max Planck really cover, if you will, very well two of the three components that shape us as human. I mean, the power in understanding human genetics, and particularly the power of gene-environment interaction coupled with a genetic is remarkable. Because as we all know, cognitive and emotional brain functions are really governed by the interaction of genetics, environment, and particularly how they work together. What I'll try to add to the mix here is really how environment really influences the outcome, and particularly environment during development. And this question is a very important question because, as you know, among early life experiences, adversity, which we define in many different ways, trauma, stress, poverty, migration, war, is really in human studies strongly associated with mental health outcome and actually also cognitive health. But I'd like to emphasize association because in human studies, it is very, very difficult to prove causality and oftentimes even more difficult to really understand the mechanism. So my lab over the past couple of decades has really focused on three different things that germane to this issue of how early life adversity may influence lifelong mental and cognitive health. First, what constitutes early life adversity? Well, I know that every single person in the room is sure that we know what early life adversity is, but maybe we don't. The other is, what are the outcomes of early life adversity? Again, we know that, and again, you know, if folks from Max Planck involved in the canonical Emory studies and inner city Atlanta studies really are the ones who set the stage to understand the outcomes of severe adversity. But we know now that this works at the population level. And I'd like to really bring to your attention a paper that came out last year that shows that at the population level, there is an outstanding predictive value of early life adversity, and you can see it here. Essentially, looking at three of the most best controlled prospective studies in human, if you look only at mental health, and okay, here's a mouse, you really see that the more adversity you have, the worse the probability of psychopathology of one sort or another. So at the population level, this works really well. But the individual level, the same studies show that there's actually very poor ability to predict. In fact, as you know, in order to have a good predictive value, you really want to have something that looks like that in a ROC curve. But the ability to predict at the individual level is almost just as good as chance. So what really are the outcomes of ELA at the individual level, and can we perhaps predict for an individual child whether they're going to be at risk for psychopathology? And second, of course, and finally, of course, what are the mechanisms by which early life adversity or ELA influences the brain? Because in order for us to do something about it, we must understand the mechanism so we can have data-driven or precision medicine, if you will. Okay, so the first take-home message, and I'll give you a final one at the end, is that really this is a pretty complex issue. And really to understand, experience, and do developmental brain plasticity that influences mental health outcome, we need a multi-level, multidisciplinary approaches, ideally across species. And again, it's really gratifying to see that all three institutions, if you will, use this approach. First, but this comes with some questions. Humans versus rodent. To what extent can rodent really recapitulate human behaviors, let alone human emotions? Second, during development, not really advancing. Okay, let's see if that works. Okay, I'm going to have to use this one. We talk about brain organizer circuits and variety of insults influencing the circuits, but actually early on in life, the circuits are not even mature. And we really need to think about how the actual maturation of the circuits and the construction of the brain is influenced by early life experience. Third, we need to realize the circuits are built of neurons, and we oftentimes think about epigenetic and other processes that maybe shape what neurons do and how they behave. But neurons actually talk to glia, and believe it or not, glia talk back. And we'll talk about this a little bit. And finally, we have a question about transient insults that happen early in life, and how do they get transmuted or transmogrified, if you will, into lifelong effects. And of course, epigenetic plays a role in that. So after this preamble, let's jump right in. So as Dominique has mentioned, we aimed to establish a way to determine causality in terms of the effects of early life adversity on outcome, and also understand the mechanism. And we came up with a very simple paradigm of, if you will, simulated poverty. For 1 week early in life, we essentially eliminate the assets of a mother in a cage, and that is the nesting material and the bedding material, and we create simulated poverty. We're very careful about doing this only through a very defined period, and we can discuss later on in terms of what that means in terms of comparing mice and humans, or rats and humans. And we know that everything, after 1 week, we return the pups back to the cage, and maternal behavior and everything else becomes normal again. So we have a defined level during a sensitive period of early life adversity. And over the years, what we've been able to show is that there are indeed enduring, causally related, enduring consequences of this. We started off with memory and cognitive function, and we found serious and progressive memory issues associated with really epigenetic reprogramming through NRSF of hippocampal neurons. What we'll talk about today, though, is more of the emotional problems, which are highly sex-specific, again, recapitulating, if you will, the human condition. In males, we find a quote-unquote anhedonia-like phenotype with reduced reward-seeking behaviors. And in females, we find the craving, if you will, of comfort food and increased motivation and addiction-like behaviors. And of course, in both sexes, altered sensitivity to subsequent stresses with epigenetic reprogramming, as we've shown in single-cell transcriptomic, of the hypothalamic neurons that are germane, and I don't think I'll have a chance to show these data. And a little bit, I will share with you poor microglial pruning of excitatory synapses. So what do we really find? Well, in male mice and rats, we see a reduction in all kinds of pleasant stuff. Sucrose preference is reduced after early life adversity during adulthood. Sexual reward cues are altered. Again, we don't really expose mice to Chanel No. 5. What we do is allow them to smell the urine of a female in heat. And they seem to be quite a bit less interested in it. And of course, palatable food. Regular food, there's no difference between the groups. This is not unique to a particular mouse strain. In fact, it's not unique to mice. We find the same thing in rats. There is no difference in overall social interaction. We're not creating, if you will, autistic rats, but there is a significant reduction in playful interaction. They just really don't care. They're miserable. In females, in contrast, we see a craving, an increased consumption of palatable food, not regular chow. Again, and this is true in both mice and rats, and is associated, interestingly, with addiction-like behaviors to drugs that are called prohedonic, or at least activate the hedonic hotspots in the nucleus accumbens. And you can see that whereas there's no increase in overall heroin intake, there is a protracted extinction and a variety of reinstatement abnormalities. And again, this is not an addiction talk, so this is for the cognoscenti, and we've published that. So both in male and females, although the phenotypes are quite different, what we see here is, at least for this type of behaviors, we're dealing with a problem with a reward secret. And again, this is just a brief diagram here, a simplified one of the reward secret in humans and in rodents. So we were really happy. Okay, we nailed this down. Now we narrow the scope to the reward secret, but where the heck do we start? I mean, you either take an army of postdocs and graduate students and say, okay, go probe using chemo or optogenetics or whatever technique you know, every single component and every single neurotransmitter in the circuit. Or, since I don't have that, you can think. And what we realize is that we're dealing with consequences of early life adversity or stress. And the brain is already designed, evolutionarily conserved, to handle this type of thing. So the variety of hotspots in the brain, if you will, that actually have the receptors to glucocorticoids, to mineralocorticoids, to sympathetic agents, and also neurons and receptors to the peptide CRH, which is really, again, conserved to handle, if you will, or modulate responses to stress throughout the brain. So with this in mind, we simply asked the question, let's go ahead and map all the CRH expressing projections to an important node, a key node of the reward circuit, and this is the nucleus accumbens. And when we did that, we found a new projection that no one has ever known about. And what I'd like to show you is this novel CRH-specific projection from the basolateral amygdala to the nucleus accumbens actually mediates, at least in males, the repressed anhedonic phenotype that I've just shown you. And first, for the experts in the room, we mapped this projection in using typical novel technology. Retrograde, we put retroviruses in the nucleus accumbens and found that the virus, the origin of these fibers is actually in the basolateral amygdala, but not in the central nucleus. Being completely obsessive compulsive, we actually verified that the virus co-expresses only in CRH expressing neurons because we use CRH iris-3 mice. We did it also anterograde, namely put stuff, constructs in the basolateral amygdala and showed that the fibers went directly to the medial shell, dorsal medial shell, where the hedonic hotspots are in the neck. We then ask, is this long-range projection GABAergic or glutamatergic? As you know, most projections are glutamatergic, and the canonic projection between the BLA and the NACs is glutamatergic, except that was not the case in our hands, because through pretty careful immunocytochemistry, I'm showing you here the same cell labeled for the retrovirus, for endogenous CRH, and for two GABAergic markers. This long-range projection is GABAergic. So it's very different from what's been known about connectivity between the basolateral amygdala and the nucleus accumbens. We also show that it's GABAergic through a variety of optically evoked currents, and what I'm showing you here is that you easily can optically evoke an iPSC, inhibitory iPSC, that goes away with pituitary, and you cannot evoke EPSC. I'm flying through this a little bit because I know this is a diverse audience, and some people will have their eyes start glazing as we talk about this. So the question is, what does this projection do? Again, the canonical glutamatergic basolateral amygdala to nucleus accumbens projection promotes hedonic behavior, promotes reward. Surprisingly enough, both chemogenetic and optogenetic activation of the projection that we discovered does exactly the opposite. In California, mice and rats do exactly what they like to do. So again, when you chemogenetically stimulate the projection in normal mice, you have reduction of reward behaviors of the 3 different reward behaviors that I showed you. And that was really kind of interesting because when you think about it, activating the projection does exactly what early life adversity does, right? These are the graphs of activating the projection on top. And these are for the same behaviors what adversity does. So that begs the question, does aberrant function, overactivity, hyperactivation of this projection really is the basis or the mediator or the mechanism of what early life adversity does to reward behaviors? Now, just to remind you, these reward behaviors are not just something we study in mice. If you believe in constructs such as RDoC and what have you, anhedonia, or reduced ability to seek and experience pleasure, is really a transdiagnostic canonical symptom and sign, actually, in psychiatry. It transcends, it's usually a harbinger of schizophrenia, very core feature of major depression, and really a key biological mechanism that may tie more than one psychiatric disorder. So the question that we ask is, okay, if indeed overactivity of this projection is a key mechanism for the long-lasting effect of early life adversity on psychiatry-related behaviors, then let's go ahead and block it, inhibit it, and see if that reverts or restores, if you will, the typical phenotype. And this is the experimental design here in which we carried out the experiment. And the answer, to a major surprise, was absolutely yes. So again, if you inhibit the projection in control mice, you get pretty much nothing. But if you inhibit it in mice that have had early life adversity, male mice, you restore, again, I'm showing you here the consumption of palatable food, you restore typical behavior, you essentially get them back to what controls is. And the fact that you get that only in early life adversity and you don't see an effect in control mice is actually kind of important. Because if we had the effects in both, it would suggest, okay, well, this projection is just a nasty projection that provokes quote unquote anhedonia or reduces reward behaviors in everybody. But the fact that it only seems to be abnormal in ELA mice is really kind of intriguing, again, suggesting that a projection within the reward circuit is specifically altered by early life adversity, at least functionally altered, disrupting reward behaviors. So this was kind of gratifying, but of course, like everything else in science, it raises a whole slew of new questions. First of all, does it work through CRH and GABA? It's got both. What are the cellular targets in nucleus accumbens? Why is it hyperactive? Is it epigenetic changes in CRH expression, GABA expression, God knows what? Did we actually modify the connectivity of the different synapses? And we don't have any answers now. You're going to have to bring me back in a couple of years to get those answers. And of course, what about females? Because I've spent some time showing you that males and females are very, very different. And we have a very talented grad student working on that now. But again, I'm afraid I don't have any answers. But the real question is, how the heck does that work? I mean, I've shown you that we could generate an aberrant connection. How do we do that? And again, I don't have the answer yet, but I have an analogous answer that might shed some light on it. So again, just to remind you, the brain is organized as circuits, okay, that work together to really execute complex behavior, which is what we are all about, as well as mice. And of course, the circuits are composed of neurons, and how neurons behave depends on genes, both the DNA, if a gene is broken, it's not going to work well, and also gene expression at the right time, the right place. But also the way that neurons hook up together is really generates emergent properties of the circuits and allow it to have its properties. But early in life, these circuits are not fully mature, and the synaptic connections are not really quite there. Yes, there are genetic components and signaling molecules that get neurons to come together and form synapses, but which ones will survive and which ones will die is really something that's activity dependent and probably during the sensitive period that we're talking about. So the question here is, might early life adversity disrupt the normal situation where some synapses are strengthened and others are lost so that you get a refined circuit? And to look at that question, we went to the, if you will, canonical fundamental type or population of cells that govern stress responses. Now, this is going to be the first and probably last talk in the history of AMH that will not show a picture of the hypothalamic pituitary adrenal circuit, because I am confident that all of you know it. So I'm not showing it. I'm simply letting you know, recapitulating the fact that in the hypothalamus and the paraventricular nucleus of the hypothalamus, there's a population of CRH expressing neurons that you see here in a CRH iris cream mouse. So this is the reporter signal, TD tomato, and this is the native immunocytochemistry using Wiley-Vale's antibody of CRH. And you can see the cells are, the mouse is pretty congruent and true to the original CRH signal. And what we found is that early in life, early adversity leads to an excess number of excitatory synapses onto these stress-sensitive and stress-regulating hypothalamic CRH cells, which alters really the circuitry. And this is something that we found initially about 10 years ago and recapitulated, and other groups have as well. So both at an anatomical level, there are too many excitatory but not inhibitory synapses onto these CRH cells. And it's only these CRH cells, not other hypothalamic cells, after early life adversity. And there's no change in any parameter of spontaneous iPSCs, but the excitatory EPSCs are augmented. So that seemed to be quite important because it is associated with altered stress responses throughout life. Again, in mice, but also this is a rather typical feature of major depressive disorder. So the question is, how does it come about? Well, we were intrigued by the fact that about 2012-13, a number of groups, particularly Beth Stevens' group, suggested that one of the mechanisms by which synapses get pruned, probably in response to activity, is through microglia. And she showed that absolutely beautifully in a visual system where there is major number of engulfment of synapses on postnatal day 5, which is a sensitive period for that particular circuit, but it's completely gone by postnatal day 9 and postnatal day 30. So we asked the question, might there be a situation here, a scenario, by which early life adversity will somehow interrupt with the ability of microglia to prune synapses? So first, Jessica Bolton in my lab asked the question, are there microglia in there early in life? And the answer was yes. And not only that, between postnatal day 4 and 8, both of them are during the sensitive period. Both of them are the period where we provide the early life adversity or typical rearing. There seem to be even more intimate relation of the microglia and neurons. Microglia are shown in green. The CRH neurons specifically are in red. So we said, great, let's go look at the microglia. And we found nothing. So both in males and females, control early life adversity, they look the same number, same density, same, I mean, you can just only imagine how many parameters we measured because we found nothing. So then we realized, okay, it's not the appearance of the microglia, it might be the function. That's where the money is. So Jessica then developed a way to label microglia in green and through a variety of crosses and neurons in red. And what you see here is a live 2-photon imaging of a thick slice, looking at the magnificent surveillance activity of microglia on neuron. So what you see here are these red CRH neurons, and the microglia send the processes and intercalate and really survey them, has been shown early in the brain. The surveillance is not just tourism. They actually check on the health of a neuron and phagocytose synapses and actually sometimes whole neurons. So I can play this again if, well, it's supposed to be playing. Anyway, so then we asked the question, is there a difference in the function, in the activity, in the mobility of microglial processes? Quantifying that was a bear, but we devised 2 different methods to do that, which I'm happy to discuss later. And regardless of which method you use, there is a reduction in the overall mobility function of microglia processes. And again, this is important because it is associated with a reduction in the engulfment of synaptic elements that you see within microglia, and actually came back to Quebec to do that using electron microscopy with Tim Lai. So these microglia really don't work very well. So looking at the molecular mechanism, we identified the receptor, which is a receptor called MERTK, and this gets into kind of really the nitty-gritty, and I'm not going to share more information with you. It is in this paper here that just came out a couple of months ago, and we describe the molecular mechanism and what have you. But the real question is, I've shown you that early life adversity creates an augmented number of excitatory synapses, and it is associated later in life with aberrant stress responses. I've also shown you that microglia don't seem to engulf synapses and don't do the work. But are these 2 lines of evidence actually causal and related? I'm sorry. Namely, does the impaired microglia activity causally causes directly is a mechanism for the altered stress responses and even for the augmented number of synapses? So to do that, we actually had to do a very complex experiment to cross a variety of mice to allow us to express inhibitory or excitatory dreads, excitatory in this case, only in microglia, and then randomize the mice, sacrifice some of them on postnatal day 10 to look at synapse number, and the others to look at behavior and hormonal responses. And the interesting thing was we had to augment microglia activity selectively without injecting mice every day with CNO, because even in California, if you inject mice every day during the first week of life, they get somewhat stressed and that messes up your experiment. So the way we did that is we created tiny little pellets of CNO. And as you can see, and I did that myself, inject the little mice here under the skin with CNO after we randomize them, as we randomize them, and just let them grow up with nobody touching them. And the result of that, again, in the paper, I'll just show you 2 salient examples. First, by activating artificially the microglia that abut the CRH cells in ELA mice, we restored a normal number of excitatory synapses. They were no longer increased. They were back to normal. Second, looking at adrenal weight, which is a very, very easy way to look at chronic stress throughout life. This is what adrenal look like in adult mice that have not had early life adversity. This is what they look like after early life adversity. And as you can see, we restored adrenal weight. We also restored behavioral responses to threat and acute hormonal responses, and that's in a paper. So what we believe is going on is normally microglia near the CRH cells survey them and prune synapses to create a normal circuit. Early life adversity, and this is selective in our case to microglia near the CRH cells, those that are elsewhere in the hypothalamus or in the thalamus couldn't care less. They do not function very well related to either neuronal or microglia signaling of the early life adversity. And that leads to an aberrant circuit and aberrant stress responses. If you artificially activate the microglia, you can restore that. So I've taken you on a little bit of a tour into the animal world, but two things I have not told you. First of all, what really are the signals from the early life adversity? What is it? How does it get into the brain, influences neuronal microglia signaling, and influences behaviors? And second, we're talking about mice, and with all due respect, we love them dearly, but their mental health is not that great, that's important. Are any of these things relevant to humans? So we began to actually look at the essence of early life adversity across species. And the idea is that in the sensitive period that we're talking about, maybe the first 2 years of life, and we can discuss why I choose these, no matter where you live, in a small village in Afghanistan or in Beverly Hills, California, the proximate environment consists of your caretakers. So no matter what happens outside in the world, the signals that the developing brain gets originate very close. So that when we think about adversity in a human or a rodent, there is a direct effect, physical or emotional, and looks like the emotional is much more important than the physical, but there's also a filtering, a buffering, a conveyance through an alteration in the behaviors of the person who directly interacts with the brain in the first 2 years of life. And we need to be cognizant of that, not only in human, but also when we do experiments. So, and this is not new. I mean, many people, particularly pioneering groups here in Morale, have considered that and looked carefully at maternal behavior. And this is therefore what we decided to do in the limited bedding and nesting cages. And we followed Michael Meany and Gid Levine's ideas and looked at the total amount of nursing. We looked at archback nursing, we looked at licking and grooming, we looked at all of the beautifully delineated important parameters. And the take-home message of this complicated slide is there's absolutely no difference. So then we realized, we really need to look a little bit better. And what I'm going to show you is a video of maternal behavior accelerated during the wake period of the mouse. This is a normal looking cage, and this is a limited bedding and nesting. Do you see a difference in the maternal behavior? Let me ask it differently. Anybody in the room cannot see a difference in maternal behavior? Okay. Eventually, this mother mouse will settle down, so the overall total amount of maternal care of any kind is going to be the same. But as you can see here, it is completely fragmented. In addition, when we used software to look at the sequences of maternal behavior, we realized that they're completely unpredictable. So again, in a normal cage, this behavior O will follow behavior C, and it doesn't make a difference what it is right now, with a probability of almost 1. On the other hand, behavior NB, which is nest building, will follow behavior SG with a probability of almost 0. So you can actually predict a sequence of behaviors. Whereas in the limited bedding and nesting cages, everything is got a probability of 0.2 or 0.3, namely everything is completely unpredictable. And we worked with our mathematicians and computational folks to actually quantify that using an entropy rate. And therefore we can really look at now not only rodent, but also human maternal behaviors. So what we seem to think here is that in the limited bedding and nesting, nothing is predictable, nothing is known. Maternal behaviors are there, but they're completely unpredictable and chaotic. And then one can ask the question, is this important? And this is really the perspective that Dominic just mentioned. Thank you, Dominic. It came out last Friday, and it seems to be generating quite a bit of buzz, because it's trying to conceptualize how early life environment may influence the building of circuits. And it starts off with really simple principles. I mean, we all know that if you cover the kitten's eye for 2 weeks in a sensitive period, two things happen. You get the Nobel Prize and the kitten will never see properly because you eliminate the normal maturation of the visual system. Ditto, Takao Hench has shown the same thing for sounds for the auditory system, but he also shown that you need patterns, you need sequences of tones to build a properly refined, a properly tuned auditory system. And Khazipov and others in Europe have shown the same thing for the somatosensory system. So the question is, can the same principles apply to emotional circuits? And again, emotional circuit maturation requires really a precise integration of all these sensory circuits and build on them. But it may very well be that if you have patterns or sequences that are not typical or predictable, you end up with a circuit that is not fully refined. Novel technology and novel optogenetics will allow us to test this in the animal, and I'm happy to discuss on how we're planning to do that in the discussion. But the one thing that I'd like to really spend a little bit of time on is really going back to the clinic, which is what I promised you in the title. Namely, are any of these things remotely important to humans? Because again, it's really important to understand mechanism in experimental systems, but we really need to be sure that they're relevant to what we do in humans. So I've been fortunate, as Dominique mentioned, to be part to lead what is known as the Conti Center, which is a large NIH-funded P50 center, which includes people from all over. This is a computational person. This is a neuroimager, an adult psychiatrist, another psychiatrist who also does basic science, a big data person, bioinformatics, you name it, psychologist. So we really try to implement the ability to ask important clinical questions, take them to the lab and come back. And I'll give you the punchline of one of our earliest studies when we focused on cognition. And the idea is that exposure to unpredictable maternal sensory signals in children influences cognitive development. And what you can see here is that in rats, we simply dichotomize them. Same thing happens in mice. We have the low predictability, which is the limited bedding and nesting cages, and the high predictability, which is a typical behavior. And we look at location memory. And of course, in children, you don't have dichotomy. You have a whole spectrum or repertoire, but we quantified entropy, and I'll show you how we do this, and again, found an important correlation. The critical thing, though, is that this correlation persisted when we covaried for everything else that we know is important in early life adversity, socioeconomic level, maternal depression, education, you name it. So these held. And they were not only true in Southern California, they were true in additional American cohort and also in Finland, where everything else is different except unpredictability. So this is a collaboration with the FinBrain group in Finland, and I really look forward to additional collaborations in Europe and elsewhere, showing that in California, which is in gold, and in Finland, which is in blue, the same correlation occur not only for cognitive function, but this is effortful for control, executive function, and emotional control that is the best predictor of how an individual will do throughout life. And this persists not only at 1 year, but also up to 9.5 years of age, which is as far as we've gone up to now. So what actually do we do? Well, we use the same principle that I've showed you in the rodent. Rather than thinking about the valence of maternal behavior, cooing and nice or slapping, we simply look at auditory, tactile, and visual behaviors. And together with the computational folks, we can generate a matrix of there's really 8 possibilities: auditory alone, tactile alone, visual alone, of course, with a child, a tuning, and a variety of combinations. And we can generate, again, a transition count and a transition probability and generate entropy just the way we've done it in a rodent. The reason I'm flying over that is because I know that you're thinking nobody can do that in a clinic. This is completely irrelevant. And you're absolutely right. So we've generated, Laura Glynn in the group, generated a questionnaire that gets a sense that is easily given in the clinic and can give you a sense of unpredictability in childhood. In fact, the name of the questionnaire is QUIC, Questionnaire of Unpredictability in Childhood. And we've just submitted a paper which distills it down even further into a mini QUIC with excellent correlation with overall QUIC and with outcome, where we can really go and ask in the real world what is going on. And I'd like to really end by, okay, before that, so what we think might be going on here is the variety of adversities that people have described are critically important. There is no doubt that they contribute significantly to outcome, again, both through gene interaction and also perhaps, in and by themselves, if you will. We're simply adding here the notion that unpredictable signals from the parent, from the home, and the community at many different time scales may be a contributor as well, because they modulate neurodevelopment and therefore can provide vulnerability to psychopathology. And whether this is real, this is true not only in well, if you will, studied small cohorts in Southern California and in Finland, whether this is real, we're now having really privileged to have the opportunity to test. We've just got a grant from the state of California that allows us to test this in, if you will, population-wide level. So what we're hoping to do here is we really have two aims. One, all physicians in California are now mandated to ask about early life adversity, about the canonical ACEs. And all they can say is they have 1 ACE, 2 ACE, 3 ACEs, and so on, which is very important, but perhaps a little frustrating because we do not really know what's going on. And as I've shown you at the beginning, at the population level, that is going to be highly predictive. But for an individual child, you don't really know which child you need to refer, which child you need to focus on, because they are at high risk of schizophrenia or what have you. So the first aim is to add the 5 questions of the mini-quick to that. And then everybody also does neurodevelopmental testing on these kids and then really see, does unpredictability actually contribute to anything in the real world? And we'll have the result in 2 or 3 years. The other is really try to see, can we identify a predictive marker for an individual child? And for this, we're going to capitalize on a pilot study in the Connie Center and recruit 120 infants and really do intra-individual metabolomics to really see if we can find a predictive value. So of course, metabolomics is something that everybody in the room knows a lot better than I do, but metabolomics really suffers from a huge amount of variance based on ethnicity, based on a billion of other parameters. What we aim to do is to eliminate this variance by taking 2 samples from the same child after a year where they either had adversity or not, and we'll know a lot about what happened this year. And then we ask the question, does the delta, does the change in methylome profile of the same child tell us something about outcome? And might it be better than a single time point metabolomics? We've piloted that in rats, where we took 1 sample of DNA from a buccal swab, so we don't have to draw blood, on postnatal day 2, then randomly assign them to either early life adversity or to normal cages, took another sample in P10, and then asked about their outcome, and we knew what the outcome is because those with early life adversity will do poorly. And a proof of principle arose, which was really informative. If you use the usual way of looking at DNA methylation, you can easily tell the effect of age. The P2 samples with principal component analysis are very different from those and from P10. But in terms of which critter had what kind of upbringing or rearing in between the 2 samples, you don't have a clue. However, if you analyze it within individual, okay, sample 2 minus sample 1, and look at the change in that sample in the methylome, you really can see, using even principal component analysis, that you can distinguish the early life adversity folks from the rats that have had a normal rearing. So we're hoping to use this, and a pilot data in the Connie Center suggests that we can actually generate profiles that will be predictive of potential vulnerabilities. Okay, so I've taken you through a whirlwind tour, and this is really, hopefully, what we came out with. First is that early life adversity directly influences adult behavior, directly meaning causally, at least in animal models. And even when we do not have gene-environment interaction to a large degree, suggesting that if you do have a genetic predisposition, it probably is much more profound. Second, the mechanism involves a disruption or interference with actually maturation of the underlying brain circuits. Third, that microglia neuron interaction and crosstalk really may play a role. I've given you an example in the hypothalamus. I would anticipate that they may be a general principle and may be true elsewhere. Finally, that signal unpredictability may very well be an additional type of adversity that we might want to pay attention to, because whereas it is something that we strive for to eliminate poverty, war, abuse, and neglect, it is a difficult task. teaching folks, teaching parents, informing people about the importance of predictability in many different timescales might be actionable, might be tractable, so it might be worthwhile pursuing. But the big message here is that going from the clinic to the lab may provide really interesting and sometimes novel insights and approaches that may be useful because we can take them back to the clinic and really enrich our understanding of human mental illness. So I will stop here and thank the many, many individuals in my lab and our collaborators around the world that really made this happen. And of course, the funding entities that somehow allowed me to do all that stuff. And you for listening. Thank you. So how does that work? Do we have time for questions?
Yes,
Questions
we
from
do.
the Zoom? Okay.
Okay.
Yes. If you have a question, just come up to the stage here so you can speak with
Gustavo.
the Zoom.
Well, thank you very much for this really top de force talk. It was, I think, really stimulating and interesting. So I think that the idea of unpredictability, it's an idea that appeals a lot to me and makes a lot of sense in understanding some of the consequences of the influence of life adversity. What I wanted to hear your thoughts on are, you show very clear sex-specific differences in outcomes, yes? Whereas the impact of this unpredictability is pre-pubertal, before the brains actually differ significantly according to sex. So how can you think, or why do you think that there are so pronounced effects when they occur at that stage?
First of all, thank you, both for the kind words and for the terrific question. I actually would like to kind of think about the premise together a little bit, because you mentioned a phrase that the brains are not that different prior to puberty. And I think emerging evidence suggests that that may not be the case. There is no doubt that puberty magnifies, amplifies the differences between a male brain and a female brain. And again, I'm talking about sexes rather than gender, so to be politically correct. But there are massive differences already at birth and probably before that. I think Peg McCarthy is one of the people who showed that in a mouse and rat, even during birth. And the idea that genes that are governed by the Y chromosome and other genes that are governed by, sex-dependent imprinting and what have you, I think that fundamentally the answer to your question is really one that you answered yourself, namely, there are massive sex differences because we are dealing with a completely different brain. Yes, the brain is going to be even more different after puberty, but fundamentally it's a different brain. Just to give you one example from actually my first science paper, which is when I was a grad student, in the mouse and in the human, it happens right after birth, there is a surge of GnRH, of LHRH, from the hypothalamus in both sexes. It goes to the sex organs, and the testis responds with testosterone, which masculinizes the brain after being converted to estrogen. The ovary does nothing. So already at birth, you have a very clear single pathway, and I'm sure there are many more, to the fact that the brains are very different. So I believe the reason we have such a profound sex difference is, one, we start off with a different brain. Second, the brain actually even gets more different later. And as you well know, many of these sex-dependent differences emerge later. When we now go to human studies and we look at infants at 1 year, 2 year, 3 years, it's difficult for us to decipher sex differences, although we already see them in a methylome in a big way. It indeed, pre-puberty and around puberty, that the behavior, the emotion differentiates, just like you said. But I think the fundamental, the foundation of these differences is much earlier. Yep.
Hi, I'm Suzanne King. I'm a psychosocial researcher here. So a lot of this was a bit over my head, but we're studying very much similar kinds of processes in my lab where we want to look at, what are the mechanisms? In our case, we're looking at prenatal maternal stress, and to try to get at the causal mechanisms, we're looking at natural disasters and looking at objectively what happened to the mothers, which is quite independent of any of their personal characteristics, versus their subjective distress or their cognitive appraisal of what was going on. So we're very much concerned with really trying to understand the causal mechanism. So my question is, so I have a graduate student who is looking at resting state functional connectivity in the brain, and also DTI, looking at structural connectivity. Based on your work, where would you predict, which network, where would you predict she would see the effects of this prenatal objective hardship?
So it's a really, really good question, and it leads to about 15 additional ones, so I'll be short. One is, I think you need to look where the money is. Namely, I would look at reward circuitry if you have a biological or a phenotypic change in, emotion and hedonia or what have you, and cognitive areas if you think you're looking at cognitive ones. But what you bring up is a question of sensitive period. And again, as we know, the brain does not develop in one fell swoop. Different circuits, different systems develop at different times, and they all have their own sensitive period. And then comparing them across species is a bit of a mess. So we actually compared hippocampus in the trans paper in one place, and now we've recently published on the reward circuitry again, trying to find out, so what is the right age to compare, humans and rodents. But I think that, and you're dealing with a prenatal brain. To me, one other thing is we are, of course, looking at functional connectivity as hard as we can, and we're finding in different places we didn't even think of. My bias would be to be really data-driven. I would look everywhere, put your seeds everywhere you can, number one, or do the hypothesis-driven based on your phenotype. Because I think that there is I would not dare to predict. So this is one of the wonderful questions where I have no answers to, and I love those more than anything else. Anybody in Zoom?
Actually, I have two questions for you, Tali. One is related to this pathway from BLA to nucleus accumbens. And I'm just wondering, did you look whether you have changes in the amount of synapses that you see in the nucleus accumbens, just the same way that you've shown for the glutamatergic cells inputs to the PBN?
It's an interesting question. So we haven't yet, Dominic. This pathway is something we've been working on over the past 2 years, I believe. And we're in submission of quote unquote the big paper. And again, it is going to open a lot of the questions. And we, first of all, need to find the target cells, because counting synapses throughout the accumbens is way above my ability. But we are now working as hard as we can on identifying the target cells. Some of them are clearly medium spiny neurons, whether they're D1 or D2. I know some of them have D1, but we need to define them. It would be even more exciting if some of them will be interneurons, because as you know, the interneurons can control hundreds and thousands of additional cells. So even a modest change in the CRH-dependent pathway will create massive changes in the neck. So once we find the target cells, I think it's a very logical thing to do to look at the number of synapses. So that's sort of the way we're going about it.
And my second question is related to the human side is okay, so predictability of parental behavior. Can you, I know you're looking at what gets to the child in terms of predictability, but can you associate the type of the characteristic of the individual parent as being a good predictable factor for what kind of care will be given? Like, you know what I mean?
Yeah. So I think you're getting at a very, very important point, namely, is the maternal behavior actually, or paternal behavior, driven by the child? Is it really something that comes out of the parent or the environment or the caretaker, or is it really all driven by the child? And if it is, then what does it mean? And if it's not, then is this then a trait? And we've studied this, at least my psychology colleagues in the group have studied this quite extensively, and we've done the same thing in the rodents. And the bottom line is that it does not seem to be driven by the child. It actually is a trait of, again, we've studied mainly mothers. And we can see that because it's true against several children in the same mother. Actually, it's true against in several litters of the same dam. And we've now actually done studies with Mar Sanchez, who studies primates, non-human primates, because some questions do a lot better with, not jumping directly into rodents where you don't have an individual pup being cared for, you have a whole litter. So that seemed to be much more of a trait of a given individual. What drives that? I do not know, probably an interaction of genetics and environment of that particular individual, but it seemed to be relatively persistent. First, if you test it at 6 months and 12 months in the controlled environment that I showed you, when we do the mathematical quantification, it's very stable. Second, it's stable across children in the same mother. So it seems to be a trait.
Thank you.
Other questions?
Well, thank you, guys. This was fun. Thank you.
Okay, so thank you very much.